Did you know that cells in your body have to regenerate and replenish themselves? Well, think about what happens to your skin in the winter. Your skin becomes dry and flakes off. What happens to those skin cells? Does your skin become so thin from loss of cells that you can see through it? Of course not. The cells regenerate to replace the ones that flaked off. This occurs through a process called mitosis. During mitosis, one cell divides to produce an identical copy of itself. If the cell is going to be identical, it must also have the same copy of DNA. DNA replication is the process where DNA makes an exact copy of itself for a new daughter cell just before mitosis. DNA replication is semi-conservative. This means that instead of having an entirely new copy of DNA for the new cell, a parent DNA strand splits in half, and each half adds a new complementary strand. The result is that each of the two double-stranded molecules of DNA are half old and half new. Let's explore how this happens. The first step in eukaryotic replication is for the enzyme helicase to unzip the double-stranded DNA molecule. The enzyme literally separates the two strands by breaking the hydrogen bonds between A and T and between G and C. Once helicase unzips the two strands of DNA, there are single-stranded binding proteins that hold the strands apart. An enzyme called primase can then put an RNA primer on the DNA molecule. The primer provides a free 3' OH so that a phosphodiester bond can be formed with the next nucleotide. DNA polymerase binds to and extends the sequence from the primer by adding complementary nucleotides. How does polymerase know which nucleotide to add? Well, if the original DNA molecule has an A, then polymerase will add a T. What do you think polymerase will add if the original molecule has a G? That's right, it'll add a C. It continues along the DNA molecule, adding the complementary nucleotide. Remember that a double-stranded DNA molecule is anti-parallel. This means if one strand is in the 5' to 3' direction, the complementary strand must be positioned in the 3' to 5' direction. Newly synthesized strands of DNA must be anti-parallel to the parent strand they are being copied from. However, the thing about DNA polymerase is that it only synthesizes new strands of DNA in one direction, only in the 5' to 3' direction. Because polymerase can only extend in the 5' to 3 direction, one strand follows along behind helicase and can extend continuously. This is called the leading strand. The other side has to wait for helicase to unzip another section of DNA so that it can lay down a primer and polymerase can extend. Because polymerase must wait for the replication fork to open, this strand is called the lagging strand and results in discontinuous fragments called okazaki fragments, named after the scientist who discovered them. Last but not least, an enzyme called ligase comes along and seals the nicks between the okazaki fragments to ensure there are no gaps in the phosphodiester backbone. DNA replication results in two identical copies of DNA that are half old and half new. The strands are complementary to one another. This new copy of DNA is ready to be placed in a new cell.